Good morning, everyone. Let's start with this one UPSC previous year question. With reference to recent development regarding recombinant vector vaccine. So we studied recombinant technology, then we studied how it can be used to make vaccines, and we studied the example of Covishield, how that whole thing works. Genetic engineering is applied in the development of these vaccines. So genetic engineering simply means that we are altering the genetic material, altering DNA. So yes, this does include some kind of alteration. So this is going to be correct. The second one is bacteria and viruses are used as vectors. So yes, bacteria and viruses both can be used as vectors. They are the ideal vectors because it's easier for us to grow them in the lab and to manipulate them. And for recombinant technology, we've seen examples of both. We used a bacteria while we were producing insulin, growth hormone, right? Similarly, we can use it for other such recombinant applications, and Covishield में हमने virus use किया था. So both of these are possible, and the answer would be C, both 1 and 2. Okay. All right. So okay. We had done cloning as our last topic, and after that we had discussed about DNA fingerprinting. Now this is another method, DNA barcoding, which is a simplified way to create data for recognizing different species of organisms. While studying the genome of different organisms, it has been identified that for every species, there is some part of its DNA which is unique to that species. It can be some cytoplasmic, some part representing the cytoplasm or anything. For different organism, we have studied and found out कि कुछ एक ऐसा part होता है जो unique होता है सिर्फ उसी species के लिए. ठीक है? Now, once we have this information, it can help us create a database which will be helpful for recognizing all the species. Suppose हमने एक ऐसा data prepare कर लिया है. Now, there is an unknown species which has been identified. Like, scientists, they will be exploring and they will be coming across new species. And a lot of times, these species, like जैसे insect है, just from the morphological features or just by its appearance, exact distinction might be difficult, okay, in every case. So we can't always be sure, but genetic material is something which is a sure thing. So हम क्या कर सकते हैं? ये unknown species मिला, you will extract the DNA of that. In this DNA, we don't need to actually do the whole genome sequencing. The whole genome sequencing is a complex process, takes a lot of time, it's a large molecule. So उसकी जगह जो भी हमें इस species के लिए चाहिए पता है that for this species or for insects this is the unique part, we will only sequence that. That part is called the barcode. You know the barcode, the payment barcodes which you have, or barcodes which take you to any links or any pages. So ये same concept है यहाँ का barcode. Barcode here means that this is something which is representative of that species, of that organism. So हमने इसका बारकोड सीक्वेंस किया, ठीक है, इसकी डीएनए सीक्वेंस की, सपोज दिस इज द सीक्वेंस CTAATC or anything, whatever is that sequence. We only need to sequence that very small segment. Now there is a database which has already been in place जिसमें सब species का information है कि अगर insects का है तो okay, the different species of insects, this is the sequence for this species, this is the sequence of this species. Now once you find a match, ठीक है, मैच मीन्स कि ये जो आपने सीक्वेंस आइडेंटिफाई किया है CTAATC, ऐसा ही एक डाटाबेस में भी है. दिस कैन बी कोरिलेटेड एंड कंफर्म दैट this is the species, and this is a sure short way for identification of species. So instead of the complete genome sequencing, we have segments called barcodes which will give the complete information. If we store these barcodes in our database, it will help us in identification of different species. Please write one line on DNA barcoding. DNA barcoding, it includes small pieces of DNA segments or barcodes which are used to identify that which organism does this DNA belong to. Which organism does this DNA belong to? Okay. Going on further, the technology with which we start today, this is called the 3-parent baby. When we studied cloning, we said that we are going to have a single parent, and that single parent is the only one whose characteristics are going to be expressed in the baby, because we were trying to make an exact copy. यहाँ पे we are saying this is a 3-parent baby. So let's see what is the logic behind this, how this will work. We are talking here about mitochondria. Remember mitochondria? What is the function of mitochondria? Powerhouse of the cell. It is the location where energy would be produced in the body. Is there anything specific about mitochondria genetically? They don't have RNA, they have DNA. But yes, the point was that they have their own genetic material, that is their special property. Mitochondria is one cell organelle which has its own DNA. The main DNA of the cell is present in the nucleus. We've been talking about that. All the chromosomes are present there. But mitochondria, जो cytoplasm में है, it also has its own DNA. And we are going to be calling this mitochondrial DNA. Or mtDNA. Now the special point of this DNA is that this DNA is inherited only from the mother. जो डीएनए हमारे न्यूक्लियस में है, for that we have been saying that both the parents contribute equally, half genetic material comes from mother, half from father. यहाँ पे ऐसा कुछ नहीं है. All the mitochondria which a person has, that means all the mitochondrial DNA, is inherited only from mother. The father does not contribute to the mitochondria in your body or to the mitochondrial DNA. If we compare the size of this mitochondrial DNA, what we find is this is a very, very small piece. Like if you have overall say 20,000 genes you have. So mitochondria पे सिर्फ 37 genes. So in comparison, it is very small. You can say it is insignificant. So it will not have any major influence. Its only job is to make sure that the mitochondria is working properly. The mitochondria whose job is to produce energy, तो वो energy producing function को ये regulate कर रहा है. That is the job of this small piece of DNA. So in a normal situation, there is nothing significant about it. I mean, which can create a variation or something. But if this is defective, that's when the problem comes. Defective mitochondrial DNA, any kind of gene defects here, can lead to mitochondrial diseases. Please keep copying all this down. Basically, these will be the diseases which will interfere with the working of mitochondria. ठीक है, mitochondria अच्छे से काम नहीं करेगा, एनर्जी नहीं प्रोड्यूस करेगा if a gene is defective here. But we are doing a lot of research these days on establishing links between mitochondria, its working, its defects, and some very common diseases also, like diabetes or Parkinson's disease. So there's a lot of research which is going on. For some diseases, we have already established the link. Like the example is Leigh disease. You don't have to go into the detail of the disease, but just know that this is a disease which is linked with improperly working mitochondria. Now, when inheritance was taking place through both the parents, then we were actually making those Mendel's cross to see कि बच्चों में disease आएगी या नहीं आएगी, or will they be carrier or will they be normal, or what is their scenario going to be. But in this case, if inheritance is only from the mother, then it is going to be a sure shot transmission. कि mother से आना है और mother defective है तो transmit होगा ही होगा. So we have to find a way to avoid this, and that avoidance can happen through a mitochondrial replacement process. We have a technology which we will call mitochondrial replacement or mitochondrial donation. We will replace the defective mitochondria with normal mitochondria. And we are going to see how that happens. This is our starting point. This is the egg of the mother who is going to have a child, but her eggs have unhealthy mitochondria, or what we can say, defective mitochondrial DNA. So you can write this in the form of a flowchart. All the steps here. The first step is you get the mother's egg, and the mother's egg has defective mitochondrial DNA. Now you have to replace this. That means कहीं और से आपको normal mitochondria चाहिए, and egg का replacement egg से ही होगा. So you bring in another donor who will give the egg, but this is a donor who is having healthy mitochondria, or you can say this is normal mitochondrial DNA. After this, we are doing the process of removing the nucleus in both of these cases. So enucleation or removal of nucleus. There are two eggs. In both the cases, you will remove the nucleus. In the case of the mother, her nucleus has her n number of chromosomes, or for human beings, the 23 chromosomes. They are the ones which will be determining the characteristics of the baby. So we keep this nucleus. From the donor egg, we don't need the nucleus. We don't need her chromosomes. We only want her cytoplasm. Cytoplasm where all the mitochondria are present. हमें सिर्फ वो चाहिए यहाँ पे. So we keep the egg without the nucleus, and then we'll do a transfer. इसका nucleus इस egg में डाल दिया, and you get a new egg. Now this is an egg which has the 23 chromosomes of the mother and the normal healthy mitochondria which have come from the donor. So mother's nucleus now in donor's egg, or you can say egg with mother's nucleus and normal mitochondrial DNA. After this egg is ready, the mitochondria have been replaced. After this, we can bring in a sperm. The sperm will come and fertilize the egg. You will get a zygote, और इससे फिर baby बनेगा. The baby who will have the properties of the egg and the sperm, the original egg, जहाँ से nucleus आया. 23 chromosomes the baby is getting from the mother's nucleus, and the other 23 will be coming from the sperm. So that is his 46 chromosomes which are determining all the properties. Along with this, you are going to have the mitochondria coming from the donor. If you compare the features, like how the baby will look like, what will be his different traits, the traits are going to come from the nucleus. जो हमारे 46 chromosomes हैं वही हमारे traits determine करते हैं, characteristics determine करते हैं. But you also have DNA from a third person. Even if it's a very small DNA, it's still DNA. And the whole point of doing this exercise was because we wanted this DNA. So to give emphasis to that DNA being used here, it is called the 3-parent baby. The third parent is the one who has donated mitochondria or mitochondrial DNA. Inheritance of DNA is basically what a parent is going to be. And though in the properties, in the features of the baby, mitochondrial small size of that DNA will not have any impact as such. But just to give significance to the fact कि ये हमने किया ही इसलिए क्योंकि हमें DNA चाहिए, इसको commonly 3-parent baby भी बोलते हैं. Is this fine? Okay. There is one more thing to add here, that is the possibility of doing this replacement at the stage of the zygote also. See, if there is an egg, okay, an egg with defective mitochondrial DNA and there is a sperm. When they fuse, there is going to be a zygote which is going to be formed. But this zygote will also have the mitochondrial defect. Its mitochondria will also be defective. Why? क्योंकि zygote में सिर्फ egg से ही mitochondria आ रहा है. That's what we have learned. कि father से कोई mitochondria नहीं आता है, कोई mitochondrial DNA नहीं आता है. So all the mitochondria in the zygote will be the same ones which have come from the mother. Okay. This gives us a possibility that we can do mitochondrial replacement at the stage of the zygote also. कैसे? अगर हम एक और एग लें और एक और स्पर्म लें, but this is an egg with normal mitochondrial DNA. Then we allow them to fertilize, we get a zygote, but this zygote will be having normal mitochondrial DNA. अब आपके पास दो zygote हैं, 2n न्यूक्लियस है, साइटोप्लाज्म है with normal mitochondrial DNA. Second case में again you had 2n number of chromosomes but defective mitochondrial DNA. At this stage also, what you can do is you can do a replacement. यहां से न्यूक्लियस निकाल के डिस्ट्रॉय कर दो. सेकेंड केस में से न्यूक्लियस रख लो सिर्फ, बिकॉज दीज आर द प्रॉपर्टीज विच यू वांट, एंड देन you can create a new zygote. So zygote में जो 2n number of chromosomes हैं वो ये parents से आ रहे हैं दोनों से, because that's the nucleus, but this now has normal mitochondrial DNA. ये किसका एग है किसका स्पर्म है, दैट विल मेक नो डिफरेंस, क्योंकि दैट न्यूक्लियस यू आर गोइंग टू रिमूव. ठीक है, तो वो किसी का भी हो सकता है. इट विल नॉट बी having any significance. The only important thing is that the mitochondria here should be normal. So we have technical terms to describe both these stages or both these types of procedures. If you are using unfertilized egg and you do the replacement in that and then fertilize it, जो first situation हमने देखी थी, it is called maternal spindle transfer. The nucleus of the egg is called maternal spindle. So that nucleus being transferred, in the case of an unfertilized egg, it's called maternal spindle transfer. The second situation is if the egg is fertilized. That means in this case of the zygote, fertilized egg is the zygote. So अगर इस केस में आप ये कर रहे हो, it is called pronuclear transfer. The nucleus of the zygote, it is called pronuclei. So this is pronuclear transfer. The main point for you to understand here is that it is possible at both stages. अगर fertilization हो जाए तो भी हम कर सकते हैं, egg में तो कर ही सकते हैं. See, any genetic changes which you want to create, or you want the genetic changes in the next generation, it is ideal to do it at the stage of least number of cells, because then you will be able to ensure कि you will be able to change all these cells. So gametes में जैसे eggs है, it's an ideal place to bring in changes, even at the stage of zygote, or like in processes like gene editing, at the early stages of the embryo where the number of cells are less, you can create such changes. They will have good results rather than doing some kind of a change in somebody like me with a billion cells in my body. So even if I have to target one particular group of cells, there are still too many cells for me to get good results. Okay. So at this stage, of course, this will work wonderfully. ठीक है? Okay. नहीं, हमने जब माइटोकॉन्ड्रिया पूरे ही चेंज हो गए तो माइटोकॉन्ड्रियल इनहेरिटेंस अब डिफरेंट होगा. ठीक है, अब जिसका माइटोकॉन्ड्रिया उसकी इनहेरिटेंस कंटिन्यू होगी. राइट, वी हैव एलिमिनेटेड ऑल द डिफेक्टिव माइटोकॉन्ड्रिया. Going on further, the next technology which we have is stem cells. Now you might have heard about stem cells or stem cells technology. The first thing would be to understand what these cells are. We'll start talking about the zygote to understand this. Zygote is the first cell of our life. Okay, zygote is going to divide and create more cells. ठीक है, आज आपकी बॉडी में जो बिलियन सेल्स हैं, the starting point of that was the zygote. It was the first cell. That's how the whole story started. This zygote could create more cells, and it could also create more types of cells. जैसे कि आपके ब्लड सेल्स अलग हैं, मसल सेल्स अलग हैं, डिफरेंट ऑर्गन्स के सेल्स अलग हैं. So all these different types also ultimately have come from the zygote. So there is a stage of cells where they have the potential to replicate themselves, the potential to create more copies of themselves, and to differentiate into different types. So like blood cell, muscle cell, fat cell, जो भी cells बनाने हैं वो बन सकते हैं. This stage of a cell, okay, be it in humans, be it in any other organisms, be it in plants, we consider this to be the stage of stem cells. So stem cells वो situation है जहाँ पे unspecialization है, and because there is unspecialization, there is a possibility of creating new cells and new types of cells. ठीक है, please write this in one line. Stem cells are unspecialized cells. Stem cells are unspecialized cells that can divide and grow into different types of cells. That can divide and grow into different types of cells. Now, the next thing we are going to talk about is what are the different properties associated with stem cells. The properties of stem cells basically means into what type or into how many types of cells can they change. So The first term we are going to use here is totipotent. As I go further into explaining what are the different types of stem cells, you'll be able to connect to this better. But just try and understand and write this also. The only truly totipotent stem cell is the zygote. Zygote जो है, it has the property of changing into all types of cells and the extra embryonic tissue. Extra embryonic tissue basically means that tissue or all those things which are needed to support the embryo. Like embryo grow karna start karega, it's going to be present in a sac, then it will have blood vessels which it will connect to the blood vessels of the mother from which it will get nutrition. Then you might know about a placenta which is present, and then there is an umbilical cord which is present. All these structures also are ultimately being formed from the zygote only. So ये zygote के ही cells हैं जो divide कर करके सब चीजें बना रहे हैं. So for zygote we can use the term it is totipotent. Okay. Let's see what are the other terms which we'll be using here. The next term is pluripotent. Pluripotent will be a type of stem cell which will form all types of cells, but not extra embryonic tissue. So इस सेल से सब टाइप्स के सेल्स बन सकते हैं बॉडी के, पर एक्स्ट्रा एम्ब्रियोनिक टिश्यू नहीं बनेगा. एंड आई थिंक यू विल बी एबल टू अंडरस्टैंड दिस लॉजिक कि एक्स्ट्रा एम्ब्रियोनिक टिश्यू बनाने की जरूरत सिर्फ जायगोट को है. No other cell in your body actually needs to do it at any stage of life. That's the only stage of life where you need all those things. So that is going to be pluripotent. इसके बाद हमारा आएगा multipotent. Multipotent will be when a cell can change into some types of cells, but not all. Some means not all. So कुछ types में change करेगा, but सब types में change नहीं कर सकता. And the last one is unipotent. This is a stem cell which changes to one type of cell only. Just to one type, one fixed type. कि अगर मसल बना रहा है तो सिर्फ मसल बनाएगा और कुछ नहीं बनाएगा, like that. So you can see as we go down, the number of cells or the different number of types of cells which are formed, that is coming down. Totipotent सब कुछ बना रहा था, pluripotent extra embryonic नहीं बना रहा है, multipotent जो है all types नहीं बना रहा है, and unipotent तो is only going to make one type. क्या हुआ? कहीं और का आया, चेक कर लेना, वापस नहीं चले जाना, किसी और जगह से आया होगा. Okay, now let's see what are the different types of cells. Types of stem cells. Basically, this means at what different stages we have stem cells available. The first stage is embryonic stem cells. When you have an embryo, at that stage, you are going to have the stem cells which are present, and they will change into all types of cells of the body. Because at the stage of the embryo, your body is still forming all the parts. ठीक है, उसको बॉडी के पार्ट्स भी बनाने हैं, उसको अंदर के ऑर्गन्स भी बनाने हैं, सब कुछ बनेगा भी. So embryonic stem cells can change to all types of cells. You can write this in one line. Embryonic stem cells exist at the early stages of life, and can form all types of cells of the body. Even after a person is born, ठीक है, एम्ब्रियो ने सारे बॉडी पार्ट्स बना लिए, completely developed, the baby is born. So even after that, there is going to be a need of stem cells in the body. We have different parts of the body undergoing different kinds of wear and tear. कोई इंजरी हो सकती है, या वैसे यूज एंड डिसयूज से भी वेयर एंड टेयर होता है. Suppose you get cut on your finger. So यहाँ पे skin disintegrate हो गई है, अंदर का blood vessel disintegrate हो गया है. You're going to lose some blood, so blood cells are going to be lost. Your body needs to have a mechanism to repair all of this. और repairing will happen through creation of new cells. आपको जो blood loss हो गया वो नए blood cells बनाने पड़ेंगे. आप जो internal organs है, जैसे liver है, liver wear out हो गया, उसके cells they are dead, so it will need new cells. You might have experienced that your skin keeps shedding every few days. The skin sheds. That means नए skin cells चाहिए skin बनाने के लिए. So in the fully developed organism also, there is a need to have a reserve of cells which can be changed to new cells whenever needed. We call these stem cells as adult stem cells. In terms of their properties, as compared to embryonic stem cells, the difference is that they are tissue specific. If I have one embryonic stem cell, I can change it to all the types. पर adult stem cells में ऐसा नहीं होगा. You have reserves of stem cells kept in different parts of your body, and they will only form that particular cell. जैसे अगर लिवर में कुछ स्टेम सेल्स पड़े हैं तो वो सिर्फ लिवर के सेल्स बनाएंगे. They will not make muscle cell or blood cell or anything like that. They will divide and produce the liver cells. If you have stem cells in the brain, they will form new brain cells. That is it, nothing else. If you have stem cells in the bone marrow, if you remember we had spoken about the bone marrow creates or forms blood cells. Okay, so bone marrow में जो stem cells हैं वो सिर्फ blood cells बनाएंगे और कुछ नहीं बनाएंगे. So there is a specificity here when it comes to adult stem cells. Please write one line. Adult stem cells are present in different parts of the body, like bone marrow, skin, brain, and they produce new cells specific to these tissues. They produce new cells specific to these tissues. Now, there is another type of stem cells, but this type is not found naturally. This is something which we have created through experimentation. So, artificially, we have produced them in the lab, and they are called induced pluripotent stem cells. This is the third and the last type of stem cells. अब induced pluripotent stem cells में क्या होता है? You take any normal differentiated cell. Differentiated means the cell who has already been assigned its function. कि अब सेल जो है मसल सेल बन चुका है या ब्लड का सेल बन चुका है, कोई सेल बन चुका है. That is what we mean when we say differentiated. It is a normal cell. So that means any cell from the body can be taken. In the lab, it can be treated. It can be induced to become pluripotent. That means it will be induced to become a stem cell. So it will lose whatever its function was. Suppose ये मसल सेल था, तो मसल सेल को लिया हमने लैब में ट्रीट किया, अब वो मसल सेल नहीं है, अब वो स्टेम सेल बन गए. ठीक है, वो मसल सेल वाला फंक्शन दैट इज लॉस्ट. We have treated it successfully. It is a stem cell now, and it is a pluripotent stem cells. Pluripotent means it can now change to all types of cells of the body. So further you can take it and it will become any type of cell of the body. So आपके पास मसल सेल था, आपने लैब में लेकर गए, उसका स्टेम सेल बनाया, and then you can make blood cell from it, you can make bone cell, you can make liver cell, pancreatic cell, whatever you want to make, because it is now a stem cell and it is a pluripotent stem cell. That means it will form all types of cells of the body. This is a very, very significant experimentation. This has also received the Nobel Prize, and this is allowing us to do a lot of different kinds of research on stem cells because of its creation. Now this also you should be able to correlate a little better with the examples. जैसे zygote को हमने बोला totipotent, क्योंकि वो all types of cells बना रहे हैं, embryonic tissue भी बना रहा है. Then you have pluripotent cells, embryo के cells pluripotent होते हैं at all their stages, and the induced pluripotent cells also same property, they can change to all types of cells. Multipotent में we are saying some types of cells. So इसका example can be something like bone marrow. Bone marrow के जो stem cells हैं, they will form all the cells of blood. Red blood cells बनाएंगे, white blood cells बनाएंगे, platelets बनाएंगे, and white blood cells also further they are of so many types, right? You have 6 types further also there, right? So all of these cells the bone marrow will be able to create, making it multipotent. But this will not form muscle cell or brain cell or intestine ka cell or any other type of cell. So there is a limitation also. That's why we are saying some types, and which sometimes that is also fixed. Unipotent में सिर्फ one type of cell. जैसे कि I have to give you an example. Suppose you have liver stem cells. The stem cells in the liver, they are unipotent because they will only form liver cells, nothing else. You can't make blood from them, you can't make bone from them, you can't make anything else. Okay, so they are limited in the sense they are tissue specific. Okay. Okay. Now the question is, think of stem cells and think of the fact that how they are giving us the capacity to create new cells. Okay, you can create any type of new cell which you want if you have the right stem cells. How do you think this can be used? What can be the applications of this? Okay, lab based मीट, ठीक है. आप सेल्स लेके जो भी मीट वाला पार्ट है वो बना सकते हो. You can get the cells from some animals. You really don't need the animal. You just take those cells in the lab and you grow it into whichever organ you want, whichever part, muscle आपको चाहिए या fat चाहिए, जो भी चाहिए उसका मीट बनाने के लिए आप वो create कर सकते हो. What else? Surgical transplantation, organ transplantation basically. That's one of the most significant applications which you can have. कि आपको कोई organ चाहिए. Normally what we do is we get an organ from another person. ठीक है, उसमें मैचिंग वैचिंग करनी पड़ती है. या फिर अब और एडवांस टेक्नोलॉजी से वी आर ट्राइंग टू टेक एनिमल्स फ्रॉम other animals also, right? There was this very successful experiment of a pig transplant. So a pig's heart, of course they used human stem cells to create that heart in the pig, induced pluripotent stem cells use kiye the, and they created that heart. The pig had that heart, and then they took that heart from the pig and gave it to a human being. And that human being went on to survive for 2 months with the pig's heart, which is one very, very, very significant achievement of recent times. Okay. We are trying to do more of such experiments also to improve the prognosis also. पर in any case, human to human transplant में तो और stem cells can be very easily used. जैसे मेरा liver अब काम नहीं कर रहा है या kidney काम नहीं कर रही है. So what can be done is my stem cells can be taken and the part of that organ which is needed that can be created in the lab. And because they are my stem cells और मेरे ही cells से नया organ बन रहा है, it's going to be a perfect match. ठीक है. Why? Because वो मेरे ही cells हैं. There's going to be no problem. The risk of rejection is going to be very low. And similarly, suppose मेरे stem cells खुद के काम नहीं कर रहे हैं, so I can use somebody else's stem cells, जहाँ पे again then matching factor will come. But उसे match करके the other person will also not need to give the whole organ, जो normal transplant में होता है. आप उसके stem cells लेके बना सकते हो जो भी part चाहिए, and then that can be given as a transplant. Anything else you can think of? See, think of this idea simply that when you have some problem, when you have some disease, the first step to managing would be a correction. कि आप दवाई देकर सही करने की कोशिश करो issue. After that, maybe through some surgical intervention, you can correct, like do a surgery and create the possibility of everything being okay again. But if that also doesn't work, the end point is giving new. ठीक है, जब रिपेयर रेस्टोरेशन नहीं हो रहा है तो रिप्लेस करना पड़ेगा. एंड रिप्लेसमेंट में स्टेम सेल्स कैन प्ले अ वेरी वेरी सिग्निफिकेंट रोल. They are cells which have the potential of dividing and changing to new types of cells. So their potential is going to be in all those areas. In all those diseases where it has reached an end point. कि अब और कुछ नहीं कर सकते हैं, so नया दे सकते हैं, नया बना सकते हैं और सब parts में ये work करेगा. Like if you look at this, this is suggesting the potentials of stem cell therapy applications. So आप किसी को brain disease है उसमें दे सकते हो. Suppose a person has Alzheimer's, is losing his memory because the brain cells have worn out. So what can be done is stem cells can be used to create new brain cells and that could be given to a person as a form of treatment. So that is going to be the perfect situation, the perfect treatment for a disease जिसमें otherwise हम कुछ खास नहीं कर सकते. Then in heart disease, in muscle disease, even in diseases like diabetes. So diabetes is because of the pancreas not producing insulin. ठीक है. So आप नए पैनक्रियाटिक सेल्स दे दो which can produce insulin, the person will not need to take insulin from outside, because now his pancreas will be producing insulin with the help of the new cells. So diabetes is going to be cured. Then bones, cartilage, different kinds of orthopedic injuries, इसमें actually काफी commonly use भी हो रहा है. Then liver की कुछ problem है, skin में burns है. There's a person whose skin is burnt and it's burnt from all areas, so there is no place left जहाँ से a graft of new skin can be given. So stem cells can be used, they can be changed into skin cells, and then that can be given as a graft. Then spinal cord, blood related disorders, कहीं पे भी आपको new cells चाहिए, so stem cells is your answer. The term which we use for this kind of treatment is, we say this is regenerative medicine. You can regenerate different parts of the body. And further, if you think of advantages here, if the same person's stem cells are used, this will mean very low risk of rejection. So a regenerative treatment available in cases where otherwise there is nothing else which you can do. Along with this, a very perfect situation of very low risk of rejection. Can you think of any other type of application? This will really change the whole research game, because you have cells which can form any part of the body and you can do real life research on them. Like suppose I'm a pharmaceutical company and I have to now create a medicine for, say, reducing acidity. Okay. Where is acid produced in the body? Which part? Stomach, right? तो मुझे stomach के लिए एक medicine बनानी है. Now, in this case, what I can do is, before actually testing on the real person, I can take stem cells, I can change them into the cells of the stomach, and with that, I will be able to have a situation where I can do all kinds of testing which I want to do. कि अच्छा ये मैंने chemical दिया which I have created. How is it interacting with the acid which is present? How much time is it taking for the stomach cells to stop producing acid? Are there any other adverse effects? So I can study all of that on real cells, on real stomach cells, which will give me very close results. It can also be used to study different kinds of diseases. जैसे suppose COVID-19, ठीक है, you have COVID-19 virus, it is impacting the respiratory system of people. But we have a lot of variants, ठीक है, अभी तक variants आते जा रहे हैं, right? You have JN1 now which is there. So I want to check that how are these different variants causing the disease differently. So I can take stem cells, I can change that into cells of the lung, I can bring in different variants of the virus and I can check कि अब ये cells के साथ कैसे interact कर रहा है, कैसे impact कर रहा है, what is happening, and that will give me very, very good results. So this use of stem cells, this can greatly revolutionize the kind of research which we are having, right? But in doing all of this, of course, advantages के point से there is this is exceptional in the sense कि this is a perfect solution to something which otherwise has no solution. लेकिन currently our problem is availability of stem cells. There is one big limitation to this process. limitations to the use of stem cell therapy, and that limitation is availability of stem cells. See the three types of stem cells which we have studied. Out of these three types, the current situation is this, that embryonic stem cells you cannot use. अगर एम्ब्रियो के स्टेम सेल आप यूज करोगे, so that means you will have to destroy the embryo. ठीक है? The baby is not going to be born if you disintegrate the embryo. So that's not allowed anywhere in the world. Research with embryos as such is not permitted anywhere. So embryonic stem cells का सीन तो खत्म. अब बचे हैं adult और induced pluripotent. This induced pluripotent stem cells, this is still at a very early stage. We are doing a lot of experimentation. We are trying to use it in different animals and see what all things we can create, and then ideas like lab grown meat and other things में use कर रहे हैं, research में use कर रहे हैं, but treatment में अभी तक बहुत ज़्यादा majorly नहीं use किए. बच गए adult stem cells. Adult stem cells, their problem is the specificity. Whatever part you want to treat, you need to take stem cells from that part. And that limitation, that those adult stem cells form only one type of cell, that is a very, very big problem. Why it is a problem? Because suppose किसी को Alzheimer's का treatment देना आपको. Now, brain में stem cells लेकर, brain से लेकर brain में डालने, which in itself is a very, very difficult process. ठीक है? So, अब blood में भी stem cells होते हैं. If we had the possibility of using those stem cells for everything, then our job would have been very easy. कि blood में से निकाला, जो बनाना है बना लो, डाल दो वापस. लेकिन अब ऐसा नहीं. The specificity is a problem that creates, or that kind of limits our options and makes the process very, very difficult. Currently, if I talk about what is the situation in India, we have different stem cell treatments which are available in some of the leading institutes in the country. But here, mostly it is the blood related disorders which we are treating. And in that also, the cost actually goes up to crores. So some very last stage diseases, we treat through these methods, and even if treatment is available, if you think the cost is going to be like 1.25 crores or 2 crores or something like that, so not everyone will be able to afford it also, right? That again creates a limitation. In terms of thinking about adverse effects, कि इसका disadvantage क्या हो सकता है, we really don't understand a lot of that. As of now, we have found very good results with stem cell therapy, and maybe when it's done on a wider scale, we'll have more understanding कि इसकी क्या-क्या long-term problems भी हो सकती हैं. But as of now, we don't really have much. We only talk about this idea of limitation in availability of stem cells. ठीक है? But we have to still recognize the power of stem cells, still recognize that how useful it could be. In the long term, this can open way for having customized treatments. अगर आप मेरे stem cells लेकर मुझे कुछ treatment दे रहे हो, so that's exclusive for me. It's not like a medicine which I'm eating, which everybody is eating the same medicine, right? It's not like that. We talk about these technologies. This is just something on a side reference which I'm mentioning, that such kind of genetic studies and genetic technologies will really create a new growth in biopharma. So pharma is medicines and treatments and therapy, but with such kind of things being linked, or with more biotechnological advancements, we can have biopharma, where biological tissue and technology can actually help us in designing specific treatment, specific therapy, specific medical procedures. Okay, so that will be biopharma. Okay. Okay. So UPSC has asked a number of questions on this, and I also have one question. ये वैसे मेंस में भी पूछा जा चुका है, prelims में भी पूछा जा चुका है. So this is a prelims question, which now you are going to solve and tell me the answer. People online, any answers? C, okay. With reference to stem cells frequently in the news, which of the following statements are correct? So I'm getting C and B online. देखते हैं. First is stem cells can be derived only from mammals. So ऐसा कुछ नहीं है, ऐसी कोई limitation नहीं है. अच्छा, left with B and C, the two answers which you have given. Okay. So at least this, that means everybody was able to identify. Okay. Stem cells can be used for screening new drugs. Yes. What do you mean by screening new drugs? Testing. Testing the new medicines. Okay. Before they are released, once you test them, that is screening new drugs. And stem cells can be used for medical therapies, right? Different kinds of treatments. So, yes. The answer will be B, 2 and 3 only. Okay. Okay. Have we written all these definitions? Biotechnology, applied biotechnology. Can you check and tell me? Yes? अच्छा ठीक है. Did we talk about twins? No, किसके लिए? कोई बात नहीं, देख लो, अपने को कोई जल्दी नहीं है. हैं? हाँ हाँ हाँ, सही है, यही है. हाँ ठीक है. ऑन बोथ ट्विन्स एंड बायोटेक्नोलॉजी डेफिनेशन दोनों मिल गए आपको? Okay, we are coming to this part when we are going to start talking about the applications. So now I am assuming that you've written the basic definition. So we'll come to this part where we are going to see the applications. See, you will be reading a lot about all this in your current affairs. So I'm just going to help you build into this topic by giving you some basic ideas. When we talk about biotechnology applications, what we see is that we can color code it in different sections based on where it is being applied. If it is being applied to aquatic life, that is going to be blue biotechnology. If it is going to be applied to agriculture, we are calling it green biotechnology. Red is medical. So जितनी भी हमने चीजें की हैं, like I told you अभी biopharma, making vaccines is also biopharma only. ठीक है. then all the different kinds of treatments, gene editing, then recombinant DNA technology, stem cell therapy, ये सब चीजें यहाँ पे आ जाएंगी रेड बायोटेक्नोलॉजी में. Then white is for industrial, and you can add one more, which is gray biotechnology, which is for environment. एनवायरनमेंट पढ़ना शुरू कर दिया आपने क्या भी नहीं? यस नो? इस पे तो किसी को कंफ्यूजन नहीं होना चाहिए. आई एम श्योर ये तो एवरीबडी वुड बी अवेयर कि हो गया है कि नहीं. अच्छा ठीक है. सो I'll talk to you a little bit about this environment thing. When you study environment, you will come across this concept again. In environment, there are basically two main problems. One is regulation of waste, second is pollution. ठीक है, यही दोनों पे the whole story of environment is going to be there, how you're supposed to be managing this. Now we have a possibility of using living systems in this way, in this process. And use of living systems or living organisms is called bioremediation. For creating remedies for pollution or waste management, we when use the living organisms, we call that bioremediation. And bioremediation can become better and more targeted if we are using biotechnology. If we can use some kind of genetic alteration to serve our purpose. एक example इसका देती हूँ आपको, very famous example. So we have this problem of oil spills. Most of the oil around the world is traveling through water bodies in those large tankers, and many a times because of some issue the oil spills. If the oil spills, it is going to be very damaging for your aquatic life, for all the plants, animals, fishes, everything which is there, and overall for the quality of water. So isko remove karna is very important, but it is a very difficult process. Removal of these oil spills is very, very challenging. So we have to find some innovative ways to do that, and one very brilliant invention is the oil zapper. Oil zapper is a group of modified bacteria. Their genetic material has been changed, and they now have the property of digesting the oil. So, ये bacteria जब oil के contact में आएंगे, they are going to digest, they are going to break down the oil, and with that the spill is going to be cleared. So such a creation of this special bacteria is going to be application of biotechnology to environment, or you can say grey biotechnology. White का भी example देती हूँ industrial setup में. Of course we can have pharmaceutical sector as one consideration, but other than that also there are a lot of chemicals and things like dyes which we can produce with the help of living organism. Think of an example where suppose you are creating a dishwashing bar. ठीक है, तो बर्तन धोने का साबुन बनाना है अब आपको. Okay, but you want this soap to have a very good property that it will easily remove all the oil, all the stickiness on the vessels. You know those ads which tell you kadi chiknai hataye minto mein, right? So that is your objective. That kadi chiknai on vessels is the cooking oil. आप oil use कर रहे हो, ghee use कर रहे हो, whatever is used, that's a little difficult to break and remove from the vessels. So what you can do is you can use something say like recombinant DNA technology and create the enzymes which break down fats. Okay, those enzymes can be put into this soap. If the soap has fat digesting enzymes or enzymes which will break down fats, the moment that soap will come in contact with all the fat is going to be broken down. And once it breaks down, it will detach from the vessel. And then it will easily flow out with the flow of water. It can easily be cleaned, right? सो कड़ी चिकनाई मिनटों की जगह सेकेंड्स में भी निकल सकती है if you have good enzymes in that, right? So innovation on such lines of different products can be wide biotechnology. As you will keep going through your current affairs, the most examples or the most references you will find in green biotechnology. Okay, so we're going to talk a little bit more about this. We will first start with the generic term, which is genetically modified organisms. So all the technologies which we have spoken about till now, from gene editing to gene silencing, recombinant DNA technology, जो भी form use करके, if we are making some changes in genetic material, that organism is going to be called genetically modified organism, or in short it is called GMOs. You can write this term in one line, please write. Genetically modified organisms. They have their genetic material. They have their genetic material altered in a way that does not occur naturally in reproduction. So the idea here is that if an organism continues to reproduce naturally, so उसमें जो भी चीजें बनेंगी, you are going to do something different. You're adding a gene or subtracting or controlling a gene, or you're giving some form of replacement of the nucleus, anything. जो भी चीज आप alter कर रहे हो, वो naturally नहीं होने वाली थी. You have intervened and changed that course. So when you do that, that is a genetically modified organism. So Nivedita is asking for a break. Nivedita, I'll definitely give you a break, but let me just finish this. उसके बाद break, पक्का. ठीक है? When we do this, or when this happens in plants, we say these are genetically modified crops or GM crops. Now the question which I want to ask you here is, if you are trying to modify crops for agriculture, what can be the objective of that? Why would you want to do it? What will be the purpose behind doing that? Shelf life increase करना. अच्छा, very good. हमने example किया भी था. Okay, higher shelf life. Ripening period increase करना accordingly. Any other qualitative feature. Pest resistance. Okay. You want that the pests should not attack your crop. Okay, lower demand of resources. All right. Okay. Good quality. I'm trying to figure out how to put that. For good quality. Quality maybe you will have to specify which feature. Higher yield. Higher yield I'll take. High yield. See, getting or taking less nutrient is not always a good thing, right? But you can maybe talk about other environmental factors that will make like more sense. अगर कोई क्रॉप ज्यादा नाइट्रोजन ले रहा है, दैट मीन्स जो फूड प्रोडक्ट बनेगा उसमें अच्छा नाइट्रोजन कंटेंट होगा, विच विल बी गुड फॉर प्रोटीन्स in your body, right? So that ideally might not always be something which is desirable. More water absorbent or less water absorbent, हाँ इस तरह के resources का advantage definitely हो सकता है. If you have something, if I'll use a generic term here, which is going to be climate resilience. Tolerant towards changes in climate, tolerant towards less water, kabhi more water, kabhi extra rainfall. So all these features, or maybe because of that, fluctuations in salinity which is there, and the soil sometimes become unsuitable to crops. So maybe we can think of crops which are more resilient to these changes. And because of this, especially in the time period of climate change, they will be more suitable for growing. Early ripening period, reduce abiotic stresses. So yes, this is about abiotic stresses. Disease resistance. We can also think of the nutritional value of our food products. Growth rate can be increased, so that is your yield only. So yeah, fortification, somebody has said. या फिर in simple terms, increased nutrients. Now, as you read more, you will come across many examples in each of these in your current affairs. So I'll also tell you a few examples. जैसे हाई शेल्फ लाइफ के लिए हमने पढ़ा था जीन साइलेंसिंग में फलवर सवर टोमेटो. पेस्ट रेजिस्टेंस के लिए we have BT cotton. We also have a variety of brinjal, BT brinjal with similar technology. High yield के लिए you have this crop currently in news, GM mustard. Climate resilience के लिए in India we have created a variety of rubber, GM mustard. Disease resistance. See, plants में the most common disease is a fungal disease. Okay, so here we have a variety of potato which has protection against blight. Blight of potato is a fungal disease. And a fortification version or increased nutrition के लिए हमारे पास है rice, golden rice, and this has additional vitamin A. So these are a number of desirable features which we can introduce to make agriculture better, to make our crops better, and we can use different kinds of technologies, the genetic technologies which we have studied, to introduce these changes. Okay. We will talk more about the challenges of this whole process, but we are going to take a break here. Okay. Then we'll come back and discuss. Now, because this is our last class, so any doubts you have from any other sections also at the end of the class, we can take that. So just look into that. So as of today, there is only one crop, one GM crop in India, which is allowed for commercial cultivation. BT cotton is the only crop which can be grown, only GM crop, which can be grown in India. बाकी जितनी चीजें हैं वो रिसर्च स्टेज पे हैं, ट्रायल स्टेज पे, एक्सपेरिमेंटेशन स्टेज पे. There is just one crop which we have allowed. Now, if you think about it, if we have allowed only one crop, that should tell you that we must have some concerns. There must be some areas about which we are concerned, because of which we are taking time or doing more research and not allowing a lot of these crops to grow. Can you think of what all concerns we can have? Okay, you can have concerns on the ecology front. क्या आएगा इकोलॉजिकल में? फूड चेन डिस्टरप्ट हो जाएगी. ओके, ठीक है. एनीथिंग एल्स? बायोडाइवर्सिटी इम्पैक्ट हो जाएगी. See, when you think of a plant, think of a crop, it's not just that one crop which will be impacted. हर क्रॉप पे कुछ ऑर्गेनिजम्स डिपेंडेंट है. एक प्लांट है, उसको कोई हर्बीवोरस एनिमल खा रहा है. देयर बी सम बीज एंड बर्ड्स डिपेंडेंट ऑन दैट. देयर बी सम माइक्रो ऑर्गेनिजम्स डिपेंडेंट ऑन दैट. देयर इज अ होल सिस्टम, राइट? सो if the crop changes, what happens to all those things? How will that be impacted? How will the environment respond to it? That is one area of concern. Along with that, the aspect of biodiversity. Biodiversity means different diverse species of one particular type of crops. Like somebody has written ignorance of native species. Okay, so how this will work? Suppose if you look at India, we might think that we have say 10,000 varieties of rice grown in different parts of the country. Now instead of that, if we decide कि नहीं, golden rice जो है वो healthy है, golden rice में vitamin A है, so we will grow only golden rice. So what will happen then is that all the other varieties, they are going to be not grown, they are going to be ignored, as Saurabh has written. And what happens to all the organisms dependent on those crops? उसका तो पूरा system disbalance हो गया. And it's not just the biotic factors, it's not just the plants, animals or microorganisms, but there is also going to be some particular way in which the soil would be working. कौन से न्यूट्रिएंट्स ले रहा है वो, कौन से न्यूट्रिएंट्स जीएम क्रॉप्स ले रहा है. देयर माइट बी अ वेरिएशन. सो सॉइल सूटेबिलिटी, वाटर यूसेज, ये सब जो फैक्टर्स हैं एनवायरनमेंट के, दे कैन ऑल बी इम्पैक्टेड. सो वन बिग एरिया विच वी आर कंसर्न्ड ऑन is going to be the environment. Environment के अलावा any other area where we have to be concerned? Fertility, soil, environment में आ जाएगा. It is correct, yeah. Consuming the crops, health basically. ठीक है, तो आप जब ये खा रहे हो क्रॉप्स, फूड क्रॉप्स में this becomes very relevant, that there can be some impact on the health of the person. Now see, look here, BT cotton approved, BT brinjal, same technology developed at the same time, did not get approval. Why? Because BT brinjal is a food crop as opposed to BT cotton. So the concerns around health are very prominently significant for a food crop which you are going to consume. And in order to be able to use it, there is a need to have a wider range of research for it. You need to have data which will explain that how this will impact the health. And that also in long-term use. जैसे suppose BT brinjal you start using and you make, बैंगन का भरता of BT brinjal. ठीक है, अब कुकिंग चेंजेस से भी आपको स्टडी करना पड़ेगा. दैट विद दिस न्यू जीन विच यू हैव एडेड, बीटी ब्रिंजल में बैक्टीरियल जीन होती है. इट इज अ रिकॉम्बिनेंट प्रोडक्ट जिसमें वन जीन हैज कम फ्रॉम द बैक्टीरिया एंड द रेस्ट ऑफ इट इज गोइंग टू बी द ब्रिंजल और द कॉटन. ठीक है, This bacterial gene is the one which is supposed to provide pest resistance, which is its main property. Now you have to study how this protein produced by the new gene, how it will change on heating, and how it will change when giving different types of temperature, right? Like the temperature for making bharta and the temperature for making, say, बैंगन का पकोड़ा, there are variable. So you need information to tell you कि हाँ, 10 years of eating BT brinjal bharta, what happens to the liver, to the kidneys. 10 years of eating BT bangan ka pagoda, how that will impact. Okay. So because of that aspect of a lot of data being needed, we are kind of concerned that how it will impact our health. More significantly for the food crops. Any other area you can think of? You'll also have concerns on the economic front. See, anything which is being prepared using technology, that's going to increase its cost. So it is going to be expensive. And because a lot of this research is happening in private sector, and because of a lot of these GM crops, they are created by different multinational companies, there is a problem of how they will be regulated. And I'll give you one example here. So there are a lot of controversies which keep coming up in around this whole idea. So एक MNC है जिसका नाम है Monsanto. This company, it produces genetically modified seeds and sells it. But what they have done is they have created these seeds with a terminal technology. Normal case में there is a crop which grows and that crop will grow, give you seeds on harvesting, and those seeds can be sown again in the next cycle. ठीक है, इस तरह से क्रॉप से सीड्स मिलते हैं जो यूजेबल है. They have created special seeds which will not produce new seeds. I mean, they will produce new seeds. जैसे आपने एक बार GM seeds खरीदे, you grow a crop, it is harvested, it gives you seed, but these seeds are terminal. They will not grow into new crops. So that means next cycle में, again, if you want the seeds, you go back to the company. This in a way creates a monopoly of this company. And it is going to increase the economic burden of the farmers who will be using these seeds. Because now nobody else can produce the seeds, they have the technology. और जो seeds निकल रहे हैं वो वैसे ही usable नहीं है, वो terminal. So such issues of economic, financial burden of GM crops is also of concern, especially for a country like India where you have a very large number of people dependent on agriculture, and most of those people are not very capable of adding or increasing their input costs because of deficiency of resources. ठीक है? You'll get to read a lot about GM crops. There is one very big issue of their regulation. Okay, that is how it is usually discussed. जैसे कि currently जो GM mustard का issue चल रहा है, ठीक है? GM mustard was ready to use many years ago, more than 10 years ago, but it still hasn't come out to be commercially cultivated. Now mustard gives us oil. So we are dependent on a lot of import of oil from the world, the cooking oil which is there, right? तो अगर ये GM mustard productivity बढ़ा देगा, so there is this understanding that we will be able to solve a lot of that problem. So that is its advantage. And what the government did was they set up a committee to review this, to conduct clinical trials, and you have this genetic engineering appellate committee. तो ये न्यूज में आता रहता है. It gave clearance, so this was ready to be rolled out, but finally this thing, somebody put in a case, it went into litigation and said कि नहीं, इससे damaging impact होगा environment पे, and then the government stopped. और अभी भी the Supreme Court is considering its merits. ठीक है, so it has been stuck in that process for many years. When you read about such controversies, you'll have to give attention to that whole regulatory system. I'll give you a little bit idea about that also. Is this it? Okay. I was supposed to have another slide. I think it's not there, but okay, I'll tell you. ऐसे ही. So you have the Department of Biotechnology. This is under the Ministry of Science and Tech. And this is the main authority deciding what or how biotechnology is going to operate. Along with this, it time and again sets up. There are some committees which are fixed committees like this one, Genetic Engineering Appellate Committee, and the government can also set up committees from time and again based on whatever requirement it has for a particular product to get approval. The challenge in this whole regulatory system is that there is a lot of duplication, there is a lot of multiplicity, you have to get clearance from different places, and we have so many acts to which a product has to agree to, right? You have Biodiversity Act, you have Drug Safety Act, you have Plant Variety Act, ऐसे बहुत सारे acts हैं. When you read about a particular issue, then the arguments will mention this also, कि इस act की ये provision है which this thing is not agreeing to. So all of that makes the process very complex and slow also for that matter. So you might have a question which might ask you to discuss what are the issues around genetically modified crops. So one of those things in general, not just for crops but for everything, is a issue of regulation. Okay, so that will need your attention. If we extend this idea overall to biotechnology, and I ask you, what are the challenges of biotechnology? Challenges means we want to grow biotechnology, we want more advancement in biotechnology, but we are not able to do that. Why we are not able to do that? What are the hurdles in that process? Can you think of anything? Lack of technology, okay. Lack of capital, okay. चलो सबसे पहले पैसा, ठीक है. फाइनेंस नहीं है, इंफ्रास्ट्रक्चर नहीं है, ह्यूमन रिसोर्स नहीं है, मैन पावर नहीं है. दैट मीन्स रिसर्च एंड डेवलपमेंट नहीं है, एंड दैट ऑब्वियसली मीन्स लेस टेक्नोलॉजी. Anything else you can further connect to this? Awareness, very, very important point. Even if we create all of this successfully, you still need to create awareness for people to understand that how this is better, or what all things they should keep in mind before use, what can be the concerns. The right form of information is very, very important here. So awareness becomes one very big challenge in promoting any kind of new technology. Sorry? रेड टेपिज्म, ठीक है, यही जो हमने पढ़ा, रेगुलेटरी इश्यूज, that how regulation is a problem, need more R&D, okay, R&D have written. इसमें एक चीज और आप ऐड कर सकते हो, लिंक बिटवीन research and industry. So currently we don't have a very good linkage between the places where research happens and all those places where actual production might take place. तो अगर कोई टेक्नोलॉजी proven created भी है तो भी it is going to take a lot of time for it to finally get into some kind of product, right? क्योंकि स्पेशली इन इंडिया रिसर्च में, यूनिवर्सिटीज में जो लोग PhDs कर रहे हैं, दे आर डूइंग समथिंग एल्स, इंडस्ट्री इज डूइंग समथिंग एल्स, so it takes a lot of time for useful research to actually be translated into useful products, right? Like one very small point I'll put in here. Just suppose somebody takes an advanced degree from a university, okay, maybe gets a PhD, super specialization, and then takes a job with a private company. The company will say that, okay, you've studied so much, very good, this was our criteria, we have selected you, अब काम कैसे करना है वो हम सिखाएंगे. Now you are under training for the next 6 months. We will train you. This is not the ideal scenario. Ideally, when somebody has spent 10 years in a particular field, he should be trained and ready to work in the field. But that's not the case because the systems, the connections are very, very different. So if we can bridge that gap, if we can create better linkages here, we'll be in a situation कि जो research हो रहा है उससे immediately product बन सकता है. Right? क्योंकि उसके बाद के भी there are a lot of steps. You still have to go through regulation. You still need to popularize that by creating awareness. ठीक है? Possible but unknown negative outcomes. Unknown negative outcomes will be covered if you have proper awareness, proper educated awareness. Affordability, yes. See, again, that can also be streamlined और पुट अक्रॉस विद द फाइनेंशियल पार्ट. गवर्नमेंट के लिए भी एक्सपेंसिव है और जिसे यूज करना है उसके लिए वर्टिकल. सी इसमें अब बहुत स्कोप है, कोई भी पॉइंट्स आप लिख सकते हो, राइट? यू शुड बी एबल टू connect it back to this, right? I am just giving you a few ideas here. Bioterrorism, issue of patents, okay, both of these things are very good points, but मैं इसको एक और extra heading में डालना चाहती हूँ, and that is the idea of ethics here. See, everything has an ethical standpoint. Okay, everything has a moral logic. Everything is some way linked to our value system of the human civilization as a whole. This for you of course is GS paper 4, right? But paper 4 में जो भी चीजें हैं वो they're not just limited to the things mentioned in the syllabus there. It can be connected to everything. And this is where UPSC is right now in terms of asking questions. So 10 years since the syllabus change, they have now exhausted all the basic things. ठीक है, जो syllabus के points से वो पूछ रहे थे. अब interconnections का time है. So interconnections ऐसे ही होंगे, हर part को किसी भी और part से connect कर सकते हैं. And in this sense, ethics is specifically something you have to think about everything's ethics. Not only will that help you if there is a question, but it will also help you get good examples to write in your ethics paper per se. ठीक है, जैसे ethics of war, यूक्रेन में war हो रहा है, उसका कुछ ethical angle है, government is bringing in some new bill, there is a speaker controversy going on in Maharashtra. Everything has a ethical angle. Technology of course has an ethical moral angle. So we need to think about that. Similar things happen in IT industry, the people who are hired. Acha. have to be trained by the firm so to be employable in the job market. Yeah. अब ये जो दो issues Veena and Vishakha have told me, first I'll write and explain this, then I'm going to ask you what other ethical issues you can get. So issue of patents here or intellectual property right, because we are talking about biological resources, I'm using this term जो आप पढ़ोगे जब environment पढ़ोगे, which is biopiracy. Biopiracy क्या है? That somebody's genetic resources are used by someone else without taking permission. जैसे for example, there is a tribe say in Lakshadweep. Lakshadweep is very famous these days, no? Okay. So there is a tribe in Lakshadweep which is, suppose, so I'm not sure what people eat in Lakshadweep, but I'll go with rice. Okay. So suppose there's a variety of rice which people in Lakshadweep are consuming. There is one particular tribe, okay, which is consuming that rice for say 800 years. Okay. Now what happens is an MNC comes, it does a genome sequencing on that rice, and it finds out that there is some protein in that rice which has very high amount of calcium. Okay, some product is there जिससे calcium बढ़ जाता है या something like that. Now what this company does is it goes and gets a patent on that. Okay, on that gene, on that product, and starts selling it. So this is the best product in the world for increased calcium amount or calcium uptake, or this company then starts selling that product all over the world and makes a lot of money. Now there are two ethical problems in this whole situation. First is they never took permission from that tribe. एक tribe जो 800 years से वो rice grow कर रही है, they never asked or seeked their permission to get a patent on their biological resource, the genetic resource which they're going to use. And the second point is that they are not going to share the benefits, the money which they are making with this tribe. Okay, so even if at some point somebody tells them कि भाई तुम्हारी तो राइस का ये प्रोडक्ट बहुत बिक रहा है दुनिया में, still those people will not be able to use it. Why? क्योंकि पेटेंट किसी और के पास. ठीक है, so this is something which is an unethical use of genetic resources. ठीक है. Ma'am, mutation in GM crops is an issue. Can we use it as a common issue for all the fields? Mutation in GM crops is an issue. See, mutation means change in genetic material जो naturally भी हो सकता है. ठीक है, जब आप खुद करोगे तो आप उसको genetic modification बोल दोगे. जब खुद से change होता है on its own in nature, then that is mutation. So genetically modified crops are mutated crops. Okay, they are, you have changed their genetic material. So I'm not exactly able to get the sense of this. Mutation in GM crops is an issue. Mutation to GM crops में होती है. That's the whole point of using them. So how exactly is that issue? You might be having that idea in your mind. I am not able to connect. Exactly. So if you want me to understand, you can elaborate a little bit more, or I have confidence that you know what you're talking about. So yes, good. Just connect it back to the topic. Okay, whenever you write an answer. Animal testing leading to animal cruelty. See, एक चीज ये आप और understand करो. We are talking about writing mains answers here. Right? That's the whole point of this discussion. Now, when you write mains answer, आपके पास एक word limit है. 150 words, 200 words, 250 words, whatever it is. And in that, usually there will be 2 parts or 3 parts of the question. ठीक है, तो अगर कोई आपसे पूछ रहा है what are the ethical issues of biotechnology, that might just be one part. जिसमें आप possibly say 80 words use कर सकते हो to write that part, because there are some other parts of the question also which you have to answer, right? अब 80 words में at the max you will be able to give 3 points, या short करके लिखोगे तो 4 points लिख लोगे, ठीक है, इससे ज्यादा का तो scope बनेगा नहीं. So you have to be very selective about what points you are picking. And of course, animal testing and cruelty of animals can be something which you can link to this kind of research, but it is not a very specific point. Specific in the sense, animal cruelty can be considered or related to consumption of meat. Animal cruelty for testing can be, for in cosmetic industry, right? बहुत सारी जगह पे this will be an issue. ये कुछ स्पेसिफिक बायोटेक्नोलॉजी का नहीं है, ठीक है, पॉइंट इज नॉट रॉन्ग, बट when you have the scope of writing only 3 points or 4 points, the relevance of that point will have all the weightage. That is how you showcase how well you understand the topic. Otherwise तो generic तरीके से तो सब आप आज भी लिख सकते हो. The fact about preparing and understanding issues is that you alleviate the content which you put across, and you should know on the idea, on a priority basis कि yes, this is more relevant to the topic, and that's what you will select. ठीक है ना? Okay. Now one more point which was given was bioterrorism. So yes, this definitely can be a good point here. Bioweapons, bioterrorism. See, when COVID-19 came, right, there were reports and a lot of speculation about how China has deliberately, created this virus so that they can wipe off the world and live alone on the planet. I mean, whatever that idea was. But ultimately, a lot of people died in China, right? So we really don't give themselves that credit. But we do understand the possibility of this happening. There is a possibility of creating a genetically modified pathogen which will be resistant to, say, all the medicines which are available right now. So आपको ऐसा bacteria बना लो which does not respond to any of the antibiotics present in the world, right? That creation is definitely possible. And if that spreads somewhere, or if that is sent as a biological weapon, it will cause diseases for which we'll have no solution. So people will die. Possibility of that kind of misuse is definitely there, which is going to be a misuse of this technology, which of course is ethically not acceptable. If a crop is created for pest resistance, after some time, it is no more resistant. If a crop is created for pest resistance, after some time it's no more resistance. Okay, so it might lose its property. So see what happens is, जैसे pests में भी changes आते रहेंगे, right? You create one kind of pest resistant crop. Now the pest itself has changed. That means ये property नहीं change होगी. ये same idea है resistance वाला, which we studied for antimicrobial resistance. कि आपने एक दवाई बनाई किसी पर्टिकुलर पैथोजन के लिए, पर पैथोजन ही चेंज हो गया, ठीक है, पैथोजन चेंज हो गया तो दवाई नहीं काम करेगी. सो यस, दिस कैन हैपन विद अस क्रिएटिंग जेनेटिकली मॉडिफाइड क्रॉप्स ऑल्सो. कि आपने क्रॉप बनाया विच विल प्रोड्यूस अ पर्टिकुलर प्रोडक्ट विच प्रोवाइड्स पेस्ट रेजिस्टेंट, बट इफ द पेस्ट चेंजेस then of course that will not work. So that will be a problem. So in that sense you are right, Saurabh. Going back to this, any more ethical conflicts or moral issues you find with this idea? Biotechnology is when we are manipulating the genetic material. We are changing the DNA of some organism. Do you think there are any moral conflicts in that, in this idea? DNA is the most fundamental thing to the existence of a particular organism, and we want to change that. The most biggest criticism of this, or the most biggest moral argument, is that this is against nature, against the natural order of things. The most fundamental thing in nature for existence of an organism is its genetic material. And if you change that, you change the whole process in which nature was supposed to be. There can be further ideas saying that if we keep doing a lot of genome editing and if we keep changing genetic material, there might be a situation where we can alter the course of evolution. How different species were supposed to transfer traits from one generation to the other, we can change that, we can alter that. That means the way organisms are supposed to evolve or change naturally, you are interfering with that. So this is like doing God's job. This is like doing God's work. And this is one of the biggest criticisms of from the ethical standpoint for biotechnology. इसमें फर्दर भी व्यूज हो सकते हैं, लाइक इन टर्म्स ऑफ डिस्ट्रक्शन ऑफ लिविंग टिश्यू. Okay, for a lot of these experiments, living cells have to be destroyed. For something like creating a successful clone, so many embryos will have to be destroyed before that becomes successful. Like we spoke about Dolly. Dolly को बनाने के लिए 200 plus embryos first died. First were unsuccessful before that became a success. So this means there is destruction of living tissue, and in case of reproductive technologies, killing of or destruction of embryos is further equated by many extreme organizations as being equivalent to death. Killing of an embryo is killing of life. So you can have destruction of living cells in all this experimentation. You can also have one more specific argument, which is that of designer babies. Do you remember designer babies? So when you do gene editing to introduce any feature, you are creating a situation where people can get customized features in their babies, right? You can all think of what all features you want in your babies, and then you can go कि ये जीन एडिटिंग करके ऐसा बेबी बना देना. ये 5 चीजें जरूरी हैं. इतनी हाइट होनी चाहिए, इस कलर की आईज चाहिए, ऐसे बाल चाहिए, ठीक है, इसको स्पोर्ट्स में डालना है मुझे, या whatever is the viewpoint. That will create a trend which is again not natural and in a way kind of frivolous alteration with the integrity of human DNA. So if this goes on happening, this can create a lot of problems which maybe we are not able to envisage also at this stage today. One more point related to the angle of economics would be the concept of equity. Polity पढ़ लिया आपने? पढ़ रहे हो polity? Equity पढ़ लिया? Equity, equality, fundamental rights. सबका साथ, ठीक है, सबका विकास. Yes, that is equity. सबका साथ and सबका विकास is equity. Okay. Equity इसमें कैसे यूज होगी? When we consider the idea of a technology, we believe that technology is for the improvement of human civilization, for the human race as a whole. But most technologies don't do that. What happens is, like, this is an expensive technology, so maybe there is a country who has all the resources, who's technologically advanced, it's very rich, and it starts making a lot of use of biotechnology. So it will have better treatment options for its diseases. It will have better crops, more nutritious crops. It will have food security. It will have better resources and everything, right from waste management to pollution control. Everything it will be able to do properly. So this means its people are going to be very, very productive now. And with that productivity, the country will be able to progress further. So एक तो पहले ही ये country बहुत आगे थी as compared to other countries of the world. Now with this, the advantages which it is getting with the use of biotechnology, that advantage further improves or further increases that gap. The gap between haves and have-nots, as we say, right? That will further increase with the benefit of the technology. So one ethical issue with the use of this technology would be that it does not really benefit everyone equally as technology is supposed to be helping. Okay, so that will be another ethical issue. Does designer baby concept consist of any life threat? See, of course, any kind of genetic manipulation which you will do, or anything which you will do in the lab, that will definitely have low chances of success. ये कोई perfect system नहीं है. Otherwise also see reproduction or creation of new organism, that's not a perfect system. Every time you have an egg and a sperm, it will not fuse and form a baby. In nature also, conception has its ups and downs. A lot of things will fail at the stage of fertilization. A lot of times egg production might not be there, sperm might not work, फिर embryo बन जाएगा तो भी fail हो जाएगा. Final stage पे baby can also die during labor. So many things can happen. So as such वो system भी perfectly 100% नहीं चलता है. So यहाँ पे तो आप alteration कर रहे हो, the rates will be further lower. ठीक है? Okay. We'll solve one question now from the prelims section. This is a UPSC prelims question. बताओ, any answers? D, B, C, A. Any answers online? B. D. Genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. What do you think about this statement? Can you make changes in eggs and sperms? Genetic changes? Yes. Or the cells which are producing them, you can do that. जैसे एक example है mitochondrial replacement which you did. Like that, any change which you introduce at the stage where these cells are supposed to be formed, those changes will be very, very well incorporated. I've told you if the number of cells are less, it's the chances of having those changes in the next generation. Of course, अगर आप किसी के eggs, cells या sperm producing cells change करोगे, तो that change will not reflected in the person, but in the next generation, right? जब वो sperm fertilize करके baby बनाएगा, उसमें वो change आएगा. But this can definitely be done. Then a person's genome can be edited before birth at the early embryonic stage. Yes? So embryo may be again same logic, less number of cells, easier to introduce changes. Now if you can agree with these two statements, your answer will be D. This question, when it was asked, there were a lot of people who did not attempt this question. The first two statements, a lot of people who have worked on their basic concepts, they were able to get to, they identified that. But the third statement looked a little off or stupid to them. And the problem with such questions is that people get very irritated when they read something they have not read. ये एक्सपेरिमेंट अभी हो गया है सक्सेसफुली, ठीक है, लेकिन जब ये क्वेश्चन आया था तब ये नहीं था, ना न्यूज में था, ना करंट अफेयर्स में था, कहीं भी नहीं था, ठीक है. सो आई थिंक दिस इज अ 19 or 20 क्वेश्चन. So human induced pluripotent stem cells can be injected into the embryo of a pig. Okay. Now what happens in general when people come across such questions जो उन्होंने बिल्कुल पढ़ा नहीं है या सोच भी नहीं पा रहे हैं, they get very irritated and then they leave the question कि यार ये क्या बात हुई, पिग पूछ लिया, ठीक है. And if that happens, you don't attempt this question, then that is also fine. The problem with such questions is that people get so annoyed with this one question कि ये तो छोड़ दिया, बाकी के अगले 3 गलत कर दिए गुस्से में कि यार ये यूपीएससी कुछ भी पूछती रहती. That is why I always emphasize on practicing a lot and being very realistic also about the pattern of questions in the exam. आपको 100 questions पूछे जाएंगे. There are going to be at the max 30 questions, 30 maximum, जिसमें आपको हर word पता होगा. Every word in all these statements, every comma, full stop, 30 maximum. बहुत इजी पेपर आ जाएगा 35, दैट्स इट. उसके अलावा जितने क्वेश्चंस हैं, देयर विल बी समथिंग यू कैन नॉट, यू नॉट रेड, या अगर रीड किया है तो याद नहीं आ रहा, ठीक है, या श्योर नहीं हो. मोस्ट ऑफ द क्वेश्चंस आर गोइंग टू बी लाइक दैट. सो यू हैव टू टेक हेल्प फ्रॉम द ऑप्शंस. द वेरी फैक्ट दैट दिस इज एन MCQ क्वेश्चन, दैट मीन्स द आंसर इज देयर. आपको चूज करना है, बनाना नहीं है. And that advantage you need to take. Mentally भी treat अपने आप को ऐसे ही करना है कि भाई मुझे eliminate करना है. You look at the design of the question. They have no option where it is 1 and 2 only. अगर उनको आपको सच में pig के बारे में पूछना होता ना, तो 1 and 2 would have been an option. But they also don't care about the pig. Right? That is why the option is 1, 2 and 3. 3 is coming automatically if you understand these very generic basic concepts. That is the idea. That is what practice will help you in. ठीक है, कि कोई बात नहीं, डरना नहीं है, आपको पिग नहीं पता, किसी को नहीं पता. Right? That's why you see cut offs अभी तो करंट ट्रेंड में 50% तक भी नहीं जा रही है. क्यों नहीं जा रही है? सबको नहीं आता कुछ कुछ, सबको सब कुछ आ भी नहीं सकता. राइट? दिस आइडिया पीपल मिस आउट ऑन, दैट इट इज फाइन इफ यू डोंट नो समथिंग. द प्रॉब्लम कम्स व्हेन यू नो समथिंग बट यू डिड नॉट पे प्रॉपर अटेंशन, or you got annoyed with this and you left this question which you could have solved if you put a little more work into looking at the options. That is the training you need to undergo. And be very realistic about your expectations. Abhi at this stage where you are at the situation where you're supposed to be practicing questions, give tests. So in that stage also, you have to keep in mind that ऐसी कोई point, if you come across also, should not really define the course of your preparation. ऐसा नहीं होना चाहिए कि आज ये स्टेटमेंट आपने पढ़ ली है तो अब मुझे पिग पे रिसर्च करना है. ठीक है, नेक्स्ट ईयर हॉर्स पूछेंगे, फिर वही प्रॉब्लम कि यार मैं तो पिग पढ़ के गया था, अभी हॉर्स पूछ लिया. ठीक है, सो दैट रियली शुडंट इन्फ्लुएंस यू, बट यू शुड फोकस ऑन द ब्रॉडर कॉन्सेप्ट्स देयर, दैट एट लीस्ट एम आई गेटिंग दैट, because that's the edge you want to take in the exam. So for this third statement, which is correct, human induced pluripotent stem cells can be injected into the embryo of the pig. ठीक है, और यही इंजेक्ट करके उन्होंने पिग में हार्ट बनाया, फिर वो हार्ट निकाल के ह्यूमन बीइंग्स को दे दिया, and that was a successful transplant also because the person lived with that heart for 2 months. Okay, and they're trying to do more such experiments to get better results. Answer here is D. ठीक है. I'm going to summarize the whole idea of preparation of this section because I personally feel this is a section which, because it is not something which everybody is comfortable with, so a lot of people will kind of feel reluctant also to prepare it, but these are the kind of sections or questions which give you that advantage, because anybody who prepares well for this exam, who puts in 1 year of work, for the prelim stage at least, it's just going to be a game of 2 or 3 questions. सब के लिए, ठीक है. ये एक जेनरिक आइडिया ही है कि वट एवर इज दी कट ऑफ, तो जिसने अच्छे से एक बार प्रिपेयर किया है वो इतना पास तो पहुंच जाएगा कि गैप फिर 2-3 क्वेश्चंस का होता है, उससे ज्यादा नहीं होता. एंड it's easier to cover up that gap if you know sections like this. So biology gives you 5 questions and you are able to solve 3 of those questions also, you are already ahead of the game for all those people who have not done that. क्योंकि standard sections तो सब लोग prepare करेंगे ही करेंगे, ठीक है. Polity, geography, history, this is, इसमें scope of variation among candidates is a little lesser. राइट. लेकिन ऐसे जो स्मॉलर सेक्शंस हैं, इसमें यू डेफिनेटली हैव एन ईजियर वे इफ यू अंडरस्टैंड द कॉन्सेप्ट्स टू गेट दैट एज. टू प्रिपेयर, ऑफ कोर्स योर दीज क्लासेस जो 7 क्लासेस हैं, दे आर डेफिनेटली द स्टार्टिंग पॉइंट. You cannot really cut short anything in this. This is already very cut short. Along with this, because you don't have any printed material and you're not supposed to be reading any books. Reading at least of basic NCERT books, I feel will be very helpful. Read the NCERT books. Whatever you are finding there, new examples, terms, उसको आप notes में लिखो, एक बार पढ़ो और then just leave those books. Okay, आप download करके digitally भी पढ़ सकते हो, whatever works for you. I'm going to repeat the list of the chapters once more in case I have missed something or you've missed something, or maybe just a final review. And I'm also adding plant chapters here. So plant portion also is helpful in understanding a lot of things, and in prelims also it can be usable in your environment section. ठीक है, तो मैं वो chapters भी बताऊंगी एक बार आपको. Class 6th, you have chapter 1 and 4. Class 7th में you have a lot of chapters 1, 2, 6, 7, 8. Class 8th, you have chapter 2, 6, 7. Class 9th में there is one chapter, but I think you should definitely study it because otherwise cell के ऊपर और कोई chapter नहीं है. This class 9th chapter is on cell. Class 10th में there is this chapter 8 which is on basics of genetics, the more simplified version, hereditary and evolution. Evolution वाला part आपका मन करे पढ़ लेना, नहीं तो छोड़ देना, but genetics वाला part आप जरूर पढ़ सकते हो. All this will make sense to you because we have studied this, so ये सब आप easily कर पाओगे. 11th and 12th is very optional. 11th you can very comfortably not study, लेकिन इसमें 2 chapters हैं, एक circulatory system पे, एक endocrine system पे. So if you wish to read a little bit more about them, then these are your chapters. Class 12th में you have all your genetics and biotech part. Okay, 2 chapters on genetics, 2 on biotech, 1 on health. So this can give you a good idea on this whole area. And one thing you have to keep in mind while approaching class 12th biology book is this is very advanced. So you cannot really read cover to cover first line of the chapter to the last line. ना समझ आएगा, ना उतना टाइम वेस्ट करने की जरूरत है. बट आप सिलेक्टिवली पढ़ सकते हो. ठीक है, आपको कोई टॉपिक पढ़ना है, आपको जीन एडिटिंग पढ़नी है, राइट, यू कैन रीड दैट, राइट. यू वांट टू सी applications of biotechnology, you can read just that portion selectively. The chapter number here is 4, 5, then 8 is on health, and then 9 and 10. one reading you must do irrespective of your background. You might have a professional degree related to biology, or you might have never studied biology. Still, this is something everybody must do. This will give you a good foundation, and you can add to this with your current affairs. Basically, the zone of questions or the topics which they will be picking up, they are going to come from current affairs. Okay, and current affairs you have to study with understanding of the concept. बेसिक इसका आइडिया इस पोर्शन को प्रिपेयर करने का यही है कि आप करंट अफेयर्स पढ़ोगे बहुत अच्छे से, बट यू विल मेक श्योर दैट यू अंडरस्टैंड एवरीथिंग दैट इज गोइंग ऑन. उसको समझने के लिए यू आर गेटिंग क्लासेस, तो अगर क्लास को देखने की जरूरत है, क्लास देखो. You feel you need an NCRT, read from the NCRT. If you feel it's still not clear, you have to do an online search, you have to do that. But everything which is there in the current affairs, which you're reading in your newspaper, that you have to make sure that you understand that properly, because questions are going to be on concepts and not really on facts. If you understand, you will be able to very wonderfully deal with it. As such, आप current affairs पढ़ ही रहे हो, वो मेहनत आप कर ही रहे हो. साइंस एंड टेक के करंट अफेयर्स में 50% इज बायोलॉजी, राइट, समटाइम्स इवन मोर देन दैट. सो ये एफर्ट इज गोइंग टू बी वेरी वेरी फ्रूटफुल. फॉर द मेंस पार्ट स्पेसिफिकली, ऑफ कोर्स करंट अफेयर्स विल बी the primary source, but a few things which you can specifically refer to can be, Vision releases this document Mains 365. ओके, सो नेक्स्ट ईयर का तो बाद में आएगा, बट अभी वाला भी जो है, इट्स देयर इज अ लॉट ऑफ थ्योरी इन दैट, सो यू कैन जस्ट गो टू द वेबसाइट एंड लुक दैट अप. ठीक है, तो उसमें पूरा बायोटेक्नोलॉजी एप्लीकेशन, स्टेम सेल टेक्नोलॉजी, क्या एप्लीकेशन है, क्या डिसएडवांटेज है, बिल्कुल वो फॉर्मेटेड आंसर पैटर्न में दे हैव गिवन. सो यू विल फाइंड सम वेरी गुड यूजेबल कंटेंट इन दिस. अभी वाला भी आप यूज कर सकते हो. इट्स कंप्लीटली रेलेवेंट बिकॉज इट इज not going to be a dated article, but general concepts on biotechnology. Then I would suggest that you look at the website of Department of Biotechnology once. Just an overview. इसमें से कुछ पढ़ना नहीं है, but the idea here is we are sitting for a government exam, right? We need to know how the government is approaching this issue. Okay, how they are dealing it. Because a lot of questions, the ideas which they are picking up, they'll pick up from the government mandate. जैसे कि क्वेश्चन था बायोटेक्नोलॉजी हेल्पिंग द पुअर सेक्शंस. ठीक है, ये यूपीएससी का क्वेश्चन है मेंस का. So something like this is what you will find as a reference on this website also. An article maybe somebody has written, or a new scheme which the government has come up. So just going through this, getting an overview will give you a good sense of what is happening. Right? इसके अलावा आप यूज कर सकते हो योजना एंड कुरुक्षेत्र. So have you heard about these magazines? Yes. Okay. So सरकार हर महीने मिनिस्ट्री ऑफ इंफॉर्मेशन एंड ब्रॉडकास्टिंग ये योजना एंड कुरुक्षेत्र रिलीज करती है. सो देयर इज अ वेबसाइट, पब्लिकेशन डिविजन ऑफ इंडिया, उस पे ये अवेलेबल है. ठीक है, यू कैन बाय इट फ्रॉम द मार्केट ऑल्सो. नाउ हाउ यू यूज दिस इज कि ऐसा नहीं कि अभी हमने को हर महीने ये पढ़ना है उठा के. ओके, बट you will find that they are on specific topics. Every month there is a new topic. Like this time it was ease of doing business. Okay. So you can just keep a tab on which topics they are picking up. And if you feel this is a topic you want to read about, this is a topic directly linked to your syllabus. ठीक है, syllabus में भी ऐसा होता है कि बहुत सारे topics हैं जिसमें already you have a lot of content. स्टैटिक में भी बहुत कुछ आ गया, करंट में भी बहुत आ गया, तो आपको लगेगा एडिशन की जरूरत नहीं है. लेकिन इट इज अ टॉपिक विच यू फील दैट ओके दिस इज समथिंग इन माई सिलेबस बट आई स्टिल हैव टू प्रिपेयर दिस, So there's no better way to prepare it than this. Again, why? Because government source है, government magazine है, high level of credibility, and some very polished experts write these. So the analysis which is given here is wonderful. The kind of examples, or if they're explaining challenges of something, they've done that wonderfully. Of course, you keep this in mind that if you pick up a Yojana or Kurukshetra, it will have like 7 to 8 articles, sometimes less or more. Fluctuation can be there, but there's going to be a lot of repetition क्योंकि एक ही टॉपिक पे है. अब ease of doing business के कितने एक variations बन सकते हैं, है ना? So for you it will be sufficient if you read 1 or 2 articles. ठीक है, एक मेन आर्टिकल पढ़ लो, उसके नोट्स बना लो, या 2 पढ़ लो, कोई कहीं लग रहा है कि टॉपिक में वेरिएशन है, दैट इज गुड इनफ. If you get the details of any scheme, or a lot of times there are a lot of suggestions given, or a lot of case studies given, or success of something, right? Then that could be something which could be usable, right? The suggestion which I will give is just make sure you know what is the topic which is there, and then selectively आप इसको use कर सकते हो. This will be a very good source. वैसे current affairs में सब cover होगा ही होगा, ठीक है? But this can be an additional source to help you. ठीक है? Okay. If you want to ask anything, you can ask, or you can send me a mail. This is my email ID, jasmine.lakshya, everything in small letters, at gmail.com. People online, anything you want to ask or tell? When did this batch start? 27th of June. Okay. And you're writing in 24? 25 wale? 25? Okay, 25 also. All right. ठीक है, 24 वालों को तो बस अब टेस्ट वेस्ट स्टार्ट कर देने चाहिए. I know I've said that before also, but again, yes, that is one big suggestion which I always have. This is a good time to start consolidating. And 25 जिनका attempt है, for them, maybe test is something optional depending on if you're in college or how much time you have. But if you're making the effort of coming to class, make sure you're not missing out on learning and revising that. क्योंकि जब तक आप attempt दोगे वो उड़ जाएगा नहीं तो सब, ठीक है? तो उसको reinforce करते रहना through regular revision. Okay, so on that note, we are going to give you an assignment question also today, which you can write. We're going to give you an exact UPSC question. So there is this question on applied biotechnology. ठीक है, वो आप असाइनमेंट क्वेश्चन में दे दीजिए. वो पूरा एग्जैक्टली यूपीएससी का क्वेश्चन है. यू माइट नीड टू सर्च एंड पुट इन अ फ्यू एग्जांपल्स इन दैट, बट जस्ट मेक एन एफर्ट इनटू राइटिंग दैट क्वेश्चन. ठीक है, ऑल द बेस्ट एंड थैंक यू.